to talk about another system in the kidney, the dopaminergic system. Although we usually think about dopamine's action in the brain as a neurotransmitter, the kidneys make their own dopamine that functions in salt excretion and blood pressure. So what is dopamine doing in the kidney, Jasmine?
Dopamine is produced by the renal proximal tubules and accounts for at least 50% of renal sodium excretion. Dopamine is made from L-dopa, which is a precursor to dopamine in the body. Enzymes converted L-dopa to dopamine in the kidney's proximal tubular cells. When there is a large amount of sodium in your body, the dopamine system works to clear it through a process called natriuresis. Would you mind explaining how dopamine actually works, Nicole?
Sure. Dopamine binds to the D1 receptor, found in the proximal tubule in the medullary thick ascending limb of Henle. When dopamine binds to the D1 receptor, this inhibits the actions of machinery that promotes the reabsorption of sodium back into the blood. More specifically, it inhibits the principal kidney sodium transporters, sodium hydrogen exchanger 3, sodium bicarbonate symporter, and sodium potassium ATPase. The dopamine system works closely with angiotensin 2, as we discussed earlier in a video. Can you talk about what happens when the D1 receptor can't respond as well to dopamine?
The G, protein-coupled receptor kinase 4, GRK4, is a gene that shuts down the actions of the hormone dopamine. It will phosphorylate the dopamine receptor, reducing the number of D1 receptors available to bind to dopamine. When the dopamine cannot bind to the receptor, the dopamine pathway cannot shut down the sodium pumps, and so more sodium is reabsorbed from the kidneys into the blood. When sodium is elevated in the body, the principal way that it is eliminated is by increasing blood pressure to induce natriuresis. This elevation in blood pressure can be harmful if not treated. Are there any other gene variants that affects proteins in the kidneys?
In the last segment, we talked about the renin-angiotensin-aldosterone system. One gene, SLC4A5, is used to make a protein, the sodium bicarbonate symporter, that works in the kidney to pump sodium back into the blood along with bicarbonate. SLC4A5 gene variants lead to excessive sodium and bicarbonate reabsorption into the blood, and thus leading to salt sensitivity and potentially high blood pressure. How does dopamine react with the renin-angiotensin-aldosterone system?
Dopamine and angiotensin 2 with the AT2 receptor having the same end result, lowering blood pressure by inhibiting the transport of sodium back into the bloodstream. The D1 receptor and the angiotensin 2 receptor are coupled, meaning one can't work without the other. The receptors both communicate with the same molecule, PP2A, through different pathways. In addition to inhibiting the transport of sodium back into the bloodstream, more D1 and A2 receptors are recruited to the cell membrane. However, the angiotensin system is always active, while the dopamine system is active when there is a high level of salt in your blood. Our study is looking at the genetic variants in GRK4 and SLC4A5. Variation in these genes affects both the dopamine system and the RAAS to lead to salt sensitivity.
Stay tuned for more information about salt sensitivity.